All right. Good afternoon, everyone. It's my distinct honor and privilege to introduce the speaker for the Paul Webster Lecture at this year's APA meeting. As you know, APA has two signature lecture in honor of two legends in the history of pancreatology. One is Frank Brook Lecture for basic science. The other one is Paul Webster for clinical and translational research. In the last 3 decades, probably, there's one speaker who has given both Frank Brook and Paul Webster lecture, if I'm correct. That is Dr. Dave Stevenson. Um, in the last couple of decades, Dave's pioneer work in pancreatic cancer biology, in understanding the interaction between pancreatic cancer cells and the microenvironment, has really shaped the field. And his first signature mouse model, we call it KRAS model, also known as Tubicin model, named after him, has really changed the way we understand the pancreatic cancer biology that shapes the landscape of pancreatic cancer, basic research, and the therapeutic resistance. And soon after that, Dave had developed a pancreatic cancer organoid system, which again has been highly influential, well organized, well accepted, and used globally. That accelerates the translation of pancreatic cancer research and therapy as well. So Dave has, the recent study on the interaction between pancreas cell and the microenvironment, carp cell. He's going to probably talk about today as well. That we really look forward to hear the groundbreaking research from Dave Stevenson, and welcome him to the stage. And Dave holds multiple leadership. I don't want to go through all of them. He's currently the Cancer Center Director at Cold Spring Harbor Laboratory, and he was AACR President 3 years ago. So Dave, take it away.
Thank you very much, Min, for your generous introduction. It's great to be back and meet with some of you. I haven't seen many of you for a while. The pancreatology field has always been very kind and considerate of oncologists like myself who attempt to peer into this tissue and organ. And hopefully I'll be able to rekindle some of my past collaborations and make some new ones. You can start the clock in the back. So I am going to talk about some recent work that we've done that will be reflected in this slide that you see here. And so this is not what it looks like when it comes out of a patient. This is processed pancreas tumor. It's actually from a mouse, from the mouse model that Min mentioned. In orange are the cancer cells, and baby blue are a type of fibroblast that I'm going to talk about today as one of the two stories. This is my disclosure slide, which won't be pertinent for today. So when pancreas cancer happens, it's an evolution of events both in the neoplastic cells as well as the microenvironment. In the neoplastic cells, you're aware of the molecular aberrations that occur with the KRAS mutation happening more than 90% of the time. And then various tumor suppressor genes get mutated along the way also. The initial step of RAS mutation is very common, probably ubiquitous throughout the population. And the first pre-invasive cancer called PANN1, also ubiquitous. Probably everybody has them. In fact, people have thousands of them in their pancreas. And yet only 1 out of 50 people get pancreas cancer, invasive pancreas cancer, diagnosed clinically in this country. And unfortunately, most of them don't survive that diagnosis. So it's a very inefficient event of progressing in the neoplastic cells. Underneath the neoplastic cells, you see the stroma. The stroma is extracellular matrix made by fibroblasts chiefly, immune cells, vasculature, etc. So there's this coevolution of the cells on top, the neoplastic cells, and the cells on the bottom, the stromal cells. And the matrix, which we and many of you have studied over many years, is supposed to be part of the healing glue of biology, to glue the tissue back together after damage. But when you have neoplastic cells, the damage never heals. It's the wound that never heals. And that material can stimulate cancer and can make it difficult for therapies to work for different reasons. So we've been studying it from that context for some time. And inflammation of the pancreas plays a really important role in all of that, the process of pancreatitis. And so approximately 10 years ago, we got involved in trying to understand the fibroblast angle. What are the fibroblasts? Where do they come from? What type of cells do they turn into? We used a model system where we first isolated stellate cells from a mouse using buoyancy gradients, immortalizing them, and then using them for various in vitro assays where you would co-culture them with organoids. In this setting, we were able to show that stellate cells could become either myofibroblasts or something we called inflammatory fibroblasts. And in the very top, you see some various pictures of that. I don't have an actual pointer, so I'll just sort of use my hands. Um, so in the very top, you see some pictures of what we called myofibroblasts. On the first picture, I don't think, yeah, the fellow said it didn't really work, but maybe it's okay. Thank you. The very first picture is where the myofibroblasts are, with the blue cells in this case. And you see a red cell farther away, that's an inflammatory fibroblast. They actually express different gene products. On the very right, you see something we call antigen-presenting fibroblasts, or AP-CAFs. These express class 2, and they have many of the hallmarks of fibroblasts. And you can find them in the in vitro models I talked about or in tissue. And the second and the third and fourth pictures are tissues. And so the concept of different types of fibroblasts came from initial studies that we initiated, and then many individuals who either independently were working on this or we had the pleasure of training in our laboratory, went on to describe other features of these fibroblasts, and many of them are listed here. So these are 7 of the probably 10 to 15 papers that have been published since then, suggesting there's more complexity besides these 3 types that we described. Well, we've been in our own way trying to understand this, taking maybe a simpler view of it, comparing health on the left and invasive cancer on the right, with stages in between of inflammation and early cancer. In this case, early cancer is just the RAS mutation. Again, this is all in a mouse, inbred C57 black 6, so it's less noisy than trying to do this in humans. We then validate everything in humans. So we took, basically a straightforward approach of using the single cell type sequencing analysis of gene expression in efforts to study all those populations. The twist is that instead of just using gene expression, we used pathway activation as the method for fingerprinting everything. And so working with a computational biologist at Columbia, Andrea Califano, he's developed various methods for looking at pathway activation, where you take sparse gene expression information and are able to construct robust pathways. And if you compare the left to the right, that's the same data, just analyzed differently. On the right by Andrea, on the left by the various data science cores that we have in our cancer centers. And so you can see it's stark, and actually the size of the populations are even different because you reassign cells. So we did this across the 4 conditions, and this is the compilation of all the data. And so you see these basically colors and shapes. Just take a look at how normal cells, meaning homeostatic fibroblasts in the pancreas, are quite similar to what you would find in chronic pancreatitis, with some exceptions. Early cancer, just the RAS mutation, very similar to pancreatitis, with some exceptions, but a huge difference when you get into an invasive advanced cancer, where you see the red cloud at 3 o'clock. And so if you get all of that data and then you computationally analyze it, you see basically 5 stages. So you had 4 conditions, but you end up with 5 stages. And the density plots on the right actually show the 5 stages mathematically. CS1, 2, 3, 4, and 5. Essentially, CS1 is cell state 1 is homeostasis, healthy mouse pancreas. Cell state 2 is pancreatitis, in this case, a cerulean pancreatitis model. Cell state 3 and 4 is actually a biphasic curve from the Pannin mice, just RAS mutation. So there's like two populations you will see. And then 5 is the invasive KPC mouse pancreas. And so that's the math. But on the left, you see all those pathways that fall out. And this is pathway activation. And I put a dotted line down the middle. So that's the Mississippi River. And if you look on the left, essentially that's health and inflammation. And on the right, that's cancer. So we'll talk about the left first and then the right second. And on the left, a couple of things came up which were a little bit surprising. The first arrow is to a track receptor. And in parentheses to the right, you see 3 of the genes that popped out in that cluster. And then the second arrow down is TGF beta activation. And actually, the ISH on the bottom in situ hybridization shows you little red dots of TGF beta transcript. You see it in chronic pancreatitis, early cancer, advanced cancer. So it starts in inflammation, persists. But we're going to talk first about the first study in nerves. So our patients with pancreatitis and pancreas cancer are unique in that they hurt. They hurt a lot. And that's what actually brings them oftentimes to attention. It's a very unusual aspect of the biology. And that actually is something that I've always thought about and always wanted to work on. And so this is our first foray into this space. Timothy Wong, who's a gastroenterologist and I suspect a esteemed member of this august society, had a couple of papers talking about how nerves can stimulate or suppress pancreas cancer, stomach cancer, and now colon cancer. Some of his findings are shown here, where sympathetic nerves in the upper right can stimulate through the beta 2 receptor activation of pathways that lead to proliferation, expansion of cancer. And on the lower left, opposite for parasympathetic nerves that use acetylcholine to somehow suppress the activation of pathways in pancreas cancer. So these are papers that were published, pre-pandemic. And then Alec Kimmelman on the lower right had a really provocative paper about how sensory nerves could feed the cancer cells, in this case, serine. So again, we had a Califano-derived finding. We then had to sort of deconvolute it and use approaches that we could do in our laboratory. And so again, using in vitro models where you can take stellate cells, pancreas stellate cells, embed them in a matrix so they can't touch, stimulate them either with TGF beta or interleukin-1, you can derive a myofibroblast or an inflammatory fibroblast phenotype and secretome. And when we got the phenotypes, we took the RNA and asked, what did the RNA show? And whenever you stimulate with TGF beta, you actually would get this axon guidance pathway popping out. And if you looked at it by Western blot, you would see that too. And so the Western to the left, this is immunoblotting for multiple proteins. If you use TGF beta, the cells produce nerve growth factor. Nerve growth factor is probably the most potent axon guidance molecule known in biology, and it's being made by fibroblasts in our case. There are other axon guidance cues there like semaphorin 7, et cetera. Turns out this whole thing is dependent on TGF beta. You're giving it, so you expect so. But if you knock it out in the blot on the right, you only see it if the receptor itself is expressed. You can take that conditioned media and stimulate murine celiac ganglia that you can isolate from separate mice and ask what would happen. And the conditioned media on the left that was stimulated with TGF beta, you get tons of neurites, tons of axons basically sprouting out compared to the conditioned media made from cells that had the knockout of the TGF beta receptor. And on the very right is a Western showing the difference in the secretomes. So again, suggestive information. But really the interesting thing is in the tissue. And so really borrowing pages from how neuroscientists study tissue and disease, you're going to see various pieces of information. So this is thick slice tissue clearing, asking questions about structure of the mouse pancreas. On the top, it's a healthy pancreas. On the bottom, it's a pancreas that has the RAS mutation. So a Pannin mouse, a KC mouse, as it were. Keratin 19 is obviously a ductal marker. It shows you the large ducts and the small ducts. Tubulin 3, beta 3 tubulin is a pan-neuronal marker. Protoplanin is a gene that's expressed by activated fibroblasts. and you see the merge. And so in the top, this is the pattern you get. On the bottom, the pattern is totally different. On the bottom, you see clusters of keratin 19. Those are individual pannins that you could see in thick slice. We estimate we have about 1 million initiating pannins in a mouse pancreas, maybe more. It's at least 1 million. And look at that wire of nerves around those pannins. And then they're all wrapped in that saran wrap of podoplanin. And so again, these are pictures from mice. And if you ask what kind of nerves are there, because tubulin 3 will pick up all nerves, majority are tyrosine hydroxylase positive. That's the enzyme that makes norepinephrine. And so the green and the red overlay, you mostly see yellow. You see a few red fibers still, and those are likely sensory nerves, but the majority are sympathetic. In the human, you can do the same experiments. You can get stellate cells from a human pancreas and ask those same questions. And when you stimulate the stellate cells with TGF beta, they make axon guidance products, the semaphorins, the NGF, et cetera. You can't get the celiac ganglion from our humans. And so we used a model system, which are rat PC12 cells, which are a historic cell line for those of you who've been doing cancer research long enough, because they're one of the cell lines that told us about transient versus constitutive activation of the RAS pathway as a process that transforms you versus causes migration. Turns out if you use TGF beta conditioned human PSC media, you get lots of neurites on the right compared to the left. So it seems that, the similar observations can be made. If you look in human tissue, this is tissue from Christine Acabozio's Donahue's lab. She's one of our best pathologists in pancreas cancer. She's very focused on early pancreas cancer as well as metastasis. So this is some tissue from some of her specimens that have pannins. We see two patterns of looking for the nerves and looking for the fibroblasts. Pattern number 1 is on top. In this case, the nerves are green, marked by the perforin, which is a protein, intermediate filament protein present in nerves. In this case, red are the cancer cells, keratin 19. And in the top specimen, you see there's some nerves near the red cells, some nerves not near the red cells. And in this case, white is smooth muscle actin. This is the overlay of the activated myofibroblasts. On the bottom is the more common pattern where you see lots of green in the white. Most of the nerves are in the fibroblast area, not in the cancer cell compartment. And these are thin sections, not thick like we had in the mouse, but nonetheless, it's a pretty prominent observation. So what about functionally? Does any of this matter? So Tim Wang previously did this experiment. If you feed a mouse 6-hydroxydopamine, this is a toxin for sympathetic nerves, kills them. It doesn't kill the cell body, kills the extensions. When you do it in our mouse models or in transplant models, basically the stroma goes away. So compare left to right. You have less mesons trichromes staining there, and actually the tumors are smaller too. So it's a similar observation to what Timothy had published previously. An interesting experiment Jeremy did, he put a calcium reporter in the same stellate cells that I was talking to you about. So this is a protein that would flash green if there was calcium burst that was released in a cell. That will happen downstream of certain receptor ligand interactions. And it turned out these were propagating calcium bursts. I'm not showing the movie, but it's pleasing. And it turned out you could then, because you have that as a signal, you can figure out pretty rapidly what receptors are you dealing with. And so he used chemical agonists and antagonists. And the agonist that recapitulated norepinephrine, which is the chief neurotransmitter of sympathetic nerves, was phenylephrine, which is an alpha-1 agonist. Doxazosin is a chemical which can inhibit alpha-1, and doxazosin was able to inhibit norepinephrine and, of course, phenylephrine. And so it looked like it was an alpha-1 process, and data I won't show you. If we used beta-specific stimulants, they don't work. And if you use beta blockers, they don't do anything. And so it looked promising that it was going to be a receptor that's not studied nearly as often. This receptor, when you look for the protein, on a normal pancreas on the very left in a mouse, is present in the capsule. There's actually a population of fibroblasts on the capsule that I'm not going to talk about today, but they express alpha-1 at baseline. When you have acute and chronic pancreatitis, early and advanced cancers, you start to see expression of this gene product in the fibroblasts that are right next to the epithelial cells that are being activated. And if you look in human specimens, pretty much the same. On the left upper and the left lower, this is the normal pancreas, you'll see expression of the alpha-1 receptor on the outside. But when you have cancer, in this case, I'm showing you cancer, you will see the thing expressed in the cells that express smooth muscle actin, which are, the fibroblasts, much more so, much more abundantly than you ever will in the neoplastic compartment, if ever. This is a Western blot showing what happens to these cells when you give phenylephrine, which is, again, a synthetic ligand cousin of noradrenaline, norepinephrine. And over a time course of, in this case, several hours, at 30 minutes, you see a spike in a protein called CREB, phosphorylation of CREB, very important integrator of calcium signaling inside cells. So that's mouse on the left and human calves on the right. So this is, a cartoon of those receptors. Alpha-1 on the left, which activates calcium signaling by activating phospholipase C. On the very right, you have beta receptors, which are, again, the common one to study. And this activates adenylate cyclase to turn on cyclic AMP production. And in the middle is alpha-2, which inhibits calcium fluxes. So again, the neuroscientists have really helped us out because they've created mice where you can shine light or give synthetic ligands and turn on these receptors and watch biology happen. Well, we use the chemogenetic method of taking a 7-transmembrane receptor and fusing to it an amino-terminal fragment that would bind to a synthetic ligand. These are called DREDs. And so we used a DRED that would activate specifically the alpha-1 signaling pathway. And it was expressed in a mouse where it was turned on in a population of fibroblasts, which we believe represents all of pancreas cancer, pancreas fibroblasts. And as far as we can tell, all of pancreas cancer fibroblasts, which is PDGF receptor alpha. And so this is using the Crelox type of technology and turned it on in this case by giving tamoxifen first, and then follow that up with stimulating the mice with cerulean several times over the course of a week, and giving them the synthetic ligand, which binds to that chemogenetic molecule that I showed you before. And in the middle, you see after the arrow, one of the curves going down acutely. That's the weight loss that you get in the mouse when you give them the synthetic ligand and pancreatitis. And if you do all the various other controls, nothing happens. So if you just do pancreatitis or just give the synthetic ligand, it doesn't matter. And so they lose a lot of weight, suggesting that there's a cooperation. If you look at the tissue after just that 1 week, look at just the top, the H&E, very different, just pancreatitis to pancreatitis with that chemogenetic stimulus on top of it. And look to the very right where you have your Mason trichrome stain, you see a lot more blue on the very right box. And then you can run through all the processes. So on the right side, you see potoplanin up in the second box. If you go down on the left side, you see tenascin C that's up. You then see lots of macrophages, lots of F480, and they're blazing with arginase. There's a lot more proliferation. And so it looks like you're hyper-activating, hyper-stimulating an inflammatory cascade by adding this chemogenetic on top of pancreatitis. What if you do it with a RAS mutation? That's this experiment. In this case, we gave the synthetic drug every week, once a week. And look at that weight loss. Boom, down, up. Boom, down, up. So it really is a powerful stimulus. The mice, we have to watch them carefully, obviously. But look at their tissue at the end of just that 3-week course. So on the very right, the mice that have a RAS mutation, have had cerulean, are getting this DKZ synthetic drug. They have a lot of fibrosis. They have a lot more pannin. The pannin are higher grade. It looks like it's stimulating, really pushing the process, consistent with what we know about pancreatitis, but doing it in an unusual way, right? We're using a neurotransmitter to, in essence, to hyperactivate the fibroblasts. So that experiment on the setting of the RAS mutation, we had to use two different recombinases. We had to use FLIP to turn on KRAS, and we had to use CRE to actually express that dread receptor. We did a end around for the cancer. We had to do a transplantation model, but we still had the DKZ system there, so the chemogenetics was present. And in that setting, when we add DKZ in the setting of a cancer cell line that we had implanted or organoid that we'd implanted, we get a lot more stroma, tumors are bigger, mice get sicker. So it suggests that, that there's potentially, in addition to nerve cancer, there may be nerve fibroblasts, interactions where it initiates through TGF beta from the epithelial cells to activate fibroblasts to become myofibroblasts, which then become a cell type which secretes neurotactic factors, bring nerves in. Sympathetic nerves for sure come in. When they come in, they can release norepinephrine. That may be a positive feedback loop, which in science and in biology are always destructive. And so we think this is an amplification cascade that we had intended to study. What about the right-hand side, which is the cancer side? Well, on the right-hand side, we saw one pathway which really just knocked us over, and that's the Wnt signaling pathway. And again, using the Califano computational wizardry, it became very clear that state 4 and state 5 was really the activation of Wnt in state 4 and turning it off in state 5. And turning it off in such a fashion that the cells now function differently, we would think. And so state 4 was where Wnt is on. You could see negative feedback loops of beta-catenin, and NKD2 is the classic example. You see these baby blue cells with the arrows around the epithelial cells. And when Wnt is turned off, you start to see expression of tenascin C, which I've always thought of as a pan myofibroblast marker, but there's obviously two types of myofibroblasts, we think. In the mouse, you start to see this show up with RAS mutation and all the time when there's RAS mutated and P53 mutated in the tumor progressing. But we don't see it in pancreatitis. There has to be a RAS mutation, and you see it stochastically then. We wanted to use it and develop an in vitro model for this. For those of you who study desmoplasia, it's always been frustrating to me that we can get these stellate cells, learn all this stuff about them, but boy, they're really not that good about making stroma. Not great. And if you just use TGF beta, you don't get a lot. It turns out if you add a little Wnt to your TGF beta, it is very powerful. And so this is just a gene expression method. And you see a lot of red on the right because you're making myofibroblasts when you have a little bit of Wnt ligand with TGF beta. And if you look at that using the Eddie Kuckerman type of method, where you lay the fibroblasts on a cover slip, gelatin coated, you can get anisotropic fibers like crazy when you have both Wnt and TGF beta present. And this is a normalized angle thing on the right. And basically, you can just see all the train tracks that you see on the very right that you don't really see in all the other conditions. This is all in mouse. We have the same information from humans. And so if we ask the question like, well, Wnt is everywhere, it's important for all development, why would we uniquely see it at this stage? So we looked for a potential ligand in the epithelial compartment and in other compartments, macrophages, et cetera. And it turned out in the epithelial compartment, there was an unusual Wnt being produced, which is Wnt7. In the mouse, 7B, and the human, 7A, and the little red arrow shows that. And on the upper right, you can see Wnt7 as these orange dots in cancers, in the preneoplastic cells. In the KPC advanced cancers, every cell has it. And on the bottom is the human. Instead of NKD2 being our marker for Wnt sensing CAFs, it's actually a collagen 12A1, but it's the same exact pattern. And on the left is actually really interesting. So not every pannin has Wnt7, but those that do always have the Wnt7 CAFs next to it. So it's strong correlation. So we then wanted to do causation, and so we have this gene. Turns out this gene is essential. Fascinating, right? So we have like cells and addition, organoids. You can't knock it out. It's crazy. And it's very important for development of the endoderm ducts in the mouse and human probably too. A very fascinating protein. It binds unusual co-receptors before it drags frizzled in, which is the classic Wnt signaling apparatus. The unconventional part is shown here, REC and GPR124. And so the Wnt7s bind to one part of REC, and you can see there's that little purple blob over there. And so we developed a way to just to loop out the little piece of peptide that would bind it, where the molecule is still present, it's just a hypomorph. The cell survived. So it obviously does additional things. The cell survived, which is important because now you can do experiments. And so we did in vitro and in vivo experiments. Here's the in vivo experiment. You put it in to a mouse, and basically they don't make baby blue cells. They don't make stroma. So you've lost your ability to do it, which is fascinating that one molecule could do that. And of course, you could think of all the in vitro experiments, and here's examples of that. We can knock down the co-receptor, in this case, GPR124, using hairpins. And when we do it, all three hairpins, essentially you make noise. You're not able to make the anisotropic fibers anymore. You can do the opposite experiment, just pump it in, overexpress it. When you overexpress it, you make a lot of fibers, as shown on the right compared to the left. And you put that in a mouse, now you have a human cancer in a mouse. Very desmoplastic, like crazy. And there's the picture on the bottom, and you can quantify all the various attributes I've been talking about. These mice metastasize like crazy too. And when they metastasize, they have a microenvironment just like the primary. Well, the title of my talk said something about medicine. I haven't said anything about medicine. Well, here's our best shot. There are bad Wnt drugs out there, which are palmitoyl transferase inhibitors called porcupine inhibitors. And I say bad because it's just hard to, you can't formulate them for humans. The ones that have been in the clinic have not worked out very well. They're fairly toxic and difficult to work with, but we can do proof of concept stuff in a mouse. And in a mouse, left compared to right on the upper, baby blue cells go away if you give them 2 months of porcupine inhibitor. And on the lower part, you've lost your stromal abundance too. What about in these advanced cancers like I've been talking about and as you've heard about? This is a dye type test, but we're using chemotherapy, adriamycin, breast cancer drug, doxorubicin, it's also called. You get red nuclei, which suggests you're getting the dye in, the drug in, when you give this porcupine inhibitor to a mouse that has a KPC tumor. And it turns out in those areas, there's a lot of problems. So this is a DNA damage marker. And so it looks like this could be a way to actually get some things into the tumor, just like 18 years ago, I claimed that we could use smooth inhibitors to do this. This is going to be a different way of doing it, and I suspect will be more functional. And so we think Wnt7 is orchestrating a different type of heterogeneity in the myofibroblast compartment. And we think it's a state of the myofibroblast. There's a Wnt sensing, and we think these others are tension forming. I just don't have time to talk about that today. That's where beta-catenin canonically is knocked down, and these cells are contracting. And again, we think it's very different from the hedgehog pathway, which we've described in the past. And we think it's related to metastasis of pancreas cancer also. And so when you think about the stroma, and you think about fibroblasts, you think about your patients with chronic pancreatitis and their pain and everything like that. I now think about things a little differently than I did before. One is, I haven't told you anything about pain yet because I'm talking about sympathetic nerves so far, but I think there's something going on with these sympathetic nerves that are getting pulled in unwittingly that are participating in the process of inflammation and neoplasia. One would posit that alpha-1 blockers may play a role in ameliorating some of this. We don't yet know the answer to that. We are pursuing it, have really no data yet. The second, though, is about this other way to make desmoplasia. We think Wnt7 is a big player in that. And so could we develop Wnt7 inhibitors, actual inhibitors? There are no good antibodies to Wnt7, which is a very curious thing since the Wnts have been around for a long time. But when you talk to the experts, it's been very hard for people to make selective antagonists to the Wnts, at least so far. But today, chemistry is so much better that I think any of these things are going to be targetable. And so the concept of drug delivery, which is on the upper right, the concept of changing the fibroblasts from activated to quiescent, which is on upper left, these are areas that I think are still rich and being pursued by many in the field. I think some of these observations we have here may contribute towards helping to sort that out. I want to show a picture of the excited people who did the work. This is a walk we do to raise money for pancreas cancer research on the South Shore of Long Island. Weather is usually terrible, but this day it was sunny, I guess. And Mojde and Jussier on the left. They're the ones who drove the WINT project. And then Jeremy is on the right, and then Sandeep next to him. These are the two who worked on the cancer neuroscience project. We have a lot of collaborators who obviously helped us with all of this, without which we wouldn't have been able to get any of this done. And this is my final slide, which are my alumni on the left and the current folks in the column 3, and a lot of the collaborators on the right. And I want to thank you for your attention, and I hope some of you will think positively about studying the fibroblasts. I think this is the area that we're ready to really go at hard. And as tools become available to genetically dissect them, and chemical and biological ways become available to interrogate and antagonize them, we potentially are going to learn and maybe help, the patients that we care about. Thank you.
Thank you, Dave. We have time for 2 questions. 2 quick questions, Emily. Anadi.
Thank you, David, for such a masterful exposition. Obviously, the stellate cells are my favorite, having been the first to discover, or at least to describe and isolate them. But I had a couple of questions about the interactions with Wnt signaling and the myofibroblasts. So do you think they are actually plastic enough to go back to another phenotype after being induced by Wnt? And the other thing is, once they're activated, the type of collagen fibers that are laid down are supposed to either inhibit or facilitate migration. So have you had a chance to look at that? And sorry, the third question is about pain, which you sort of touched upon. But in terms of the interactions with these myofibroblasts with sensory neurons.
Yeah, so thank you very much for your different perspectives and questions. What we do know is that there's reversibility of the acute increase in fibrosis. What we don't know is if the cell survives and becomes less differentiated, as opposed to it dies and is replaced. So that's a fundamental question we haven't been able to address yet. The motility and the biomechanics we're extremely interested in this topic. I wouldn't say we have a lot of data yet, but the myofibroblast where the classic beta-catenin pathway is turned down, express lots of genes and have a structure of a cell that's much more contractile than do the Wnt sensing, which essentially are factories of matrix production. And these, we hypothesize that these are the cells that are actually probably moving around more. And maybe they're the ones that are, contracting. One question I thought you were going to ask me is, which type of fibroblasts talk to the nerves? And you would think I would know this. And I don't exactly know. I think maybe they both talk to the nerves. It would be much more pleasing if there was only one of the two types that talk to the nerves. But I think they both express NGF, and they both have the other axon guidance factors. And then lastly, your question about pain, I don't know yet. But we're hopefully gonna work on that. But it'd be fun to catch up. Yeah, just please.
Hi, Jamie Salomon, pain and cancer neuroscientist. So my question follows up a little bit on that. While we're trying to still figure out which subtype of fibroblast to be targeting, since they do release NGF, and drugs targeting and biologics targeting NGF have been in clinical trials for pain conditions. As a clinician, is there a path to test that in our cancer patients?
Yeah, so that's a terrific question. And we're trying to do that using genetics right now. And it looks promising so far. And so that if it really pans out, we're going to go right to small molecules, which do exist, are actually approved, et cetera. So a really spot on question. Because that actually could be something that could be used in the clinic for people who have pancreatitis or at risk for pancreas cancer, et cetera. So it's a great question. Great question.
All right. Thank you so much, Dave, for the awesome talk. As always, every time I hear your presentation, learn a bit. Hold on a second. We have something for you. And we really, the field really appreciate all the pioneer work you have laid down. You have so many model systems you have developed, from KPC model to the organoid and so on. And I see many of your trainees are here in the audience. You have trained many people who become leading expert in the field already. So AP has some appreciation for you. I want to invite our president.
You know what, I'll actually go up here.
Okay,
and
all right.
present it formally. All right, let's see if we can actually get this out of here. All right, so this says Paul Webster Lecturer, Distinguished Speaker, Dr. David Tuvson, in appreciation of your participation, APA 25 Annual Meeting, November 2025. So thank you and congratulations.